Assalamualaikum and good morning. Can you guys hear me? Hello?
Hello.
Hello?
Boleh dengar doktor?
Wah, ingat tak ada orang. Okay, kita tunggu. We wait until everybody join the class. Right now only 35. Can you see the presentation?
Yes.
Okay.
Okay.
Okay, thank you. Why your voice seem like you just wake up from your sleep? Where is the other guys? 9 of you still not registering. Six more. Another 4. Not yet. Join the class. So should we proceed? Hello.
Yes, doktor.
Where's everybody? Okay, guys. We start our class.
Okay, doktor.
Okay, doktor.
All right. Today class is on SBP 3006, Laboratory Sciences and Instrumentation, right? And the chapter is on plant extraction. Okay. So basically, in our department, most of the lecturer are doing on research on plant, on medicinal plant or medicinal potential of a plant. Okay, basically most of the lecturer in our department. Since we join with genetics lecturer, the genetic lecturer they are more on patient-based research, But the real biomedic, most of them are doing on mostly on plant extraction or plant-related bioactive compounds. Yeah. So it is an important subject for you also, that you, so that when you go to your, especially when you go to your final project, you need to know what type of plant extraction that you need to do, why you need to do this kind of extraction, not that kind of extraction, what is your lecturer's target. So there are various type of extraction, okay, to get the bioactive compound. For example, morphine. Morphine is a pure compound. It comes from a plant called Papaver somniferum, if I'm not mistaken. Okay, so we want to isolate the pure compound, so that the pure compound, if you use a mixture, if you use a crude extract, for example, it contains a lot of compounds. So if you want to target a disease, it can also have side effect, because of the action of various type of compounds. So that's why when researcher doing research on medicinal plant, the aim at the end of their study is to isolate the bioactive compound, to get a pure compound. So normally when you do research, you need to collaborate with a chemist, medicinal plant chemist. Okay. So what I'm trying to say is that when you do research, when you go out there, when you do research, if you are a lecturer or researcher at FRAME, at any institute like nuclear agency, like CRAM, whatever, MARDI, if you're doing on research, you need to collaborate. You cannot do research alone. Okay? So you have your own specialty. Later, when you do your master, you do your PhD, you become an expert in one field, but you are not expert in the other field. For example, when you do like me, I'm doing master and PhD in pharmacology on medicinal plant, but I'm not an expert in chemistry part. Okay, so I need to collaborate with researcher who's doing on chemistry part. I need to collaborate with researcher who is doing on in vitro works, like what we have learned in toxicology class, because they are more expert on how to grow the in vitro cell. Me, I'm on in vivo actually. My work is more on in vivo, using animals. You take the organ, you measure certain parameters to using the animal itself, the behavior. You take the organ to support your and look at the histology and so on. That is my part. Then you need to do in vitro study also to further confirm what you have obtained using in vivo models. So, okay, we go back to plant extraction. Okay. So, if this is for men, most of the men, they like to have a big muscle body. So, are there any plants that can if you found a plant that can help to build muscle body, you can become a rich person, no? So basically, why you need to do extraction, it is to separate a medicinally active compounds or portions of plants, what you need to have is you need to use a solvent. The solvent whereby you put your plant, you soak your plant with that solvent, and then the things that come out from the plants into the solvent is taken up and used for further isolation process. Uh, for- this is, for example, you put your plant, your dried leaves, your dried bark, or any part of the plant that you are studying, the fruit or the stem, whatever it is, you put in a solvent. It can be water, it can be organic solvent, for example, methanol, ethanol, chloroform, depending on what you want to isolate out the bioactive compound, the nature of the bioactive compound. Let's say you are targeting uh, the water-based bioactive compound, so you need to use water. Okay, you need to soak your plant in water for how many hours or how many days, depending on your procedure. Okay, so if you are just a researcher who don't care, sometimes when you do research, like for me, I'm trying to isolate a bioactive compound based on traditional use. Okay, sometimes some researcher, they don't care whether it is traditional use or not. They just want to isolate the bioactive compound. So if you want to, for me, if you want to isolate a compound from based on traditional use, that means in traditional use, normally we use water, right? And in some cases, like for example, in Chinese traditional herbs, they use alcohol to soak the dried leaf or whatever part of the plant. So it is called tincture, and you can drink it as alcohol-based medicine. Okay, that is the only two. If you want to know about traditional medicine, there are only two type of solvent used: only water and ethanol. Okay, you cannot use methanol because methanol is highly toxic. Only water and ethanol. But in some cases, researcher will tend to go for water because water is free, easy to get, compared to ethanol, which is 100, around 100 ringgit per bottle, okay, which is expensive. Unless you get a big research grant, you can buy ethanol you can use ethanol as your solvent. But normally we use water to mimic the direct effect of traditionally used medicinal plants. Any question? Hello.
No, doctor.
No,
Okay,
doctor.
clear? Boleh?
Boleh,
Boleh.
doktor.
Boleh.
All right. Please respond as soon as possible. Okay. Okay, the product. So when you do extraction, what you will get is, let's say you put a sachet of tea, tea bag, you put in a cup of water, then you see something brownish come out into the water, right? So that is actually a complex mixture of metabolite, a complex mixture of bioactive compounds. Okay. So what you drink, let's say you drink a tea, it is a mixture of compound that goes into your body. Okay. So that's why sometimes you can get, if you some tea like product, they claim to have high antioxidant, but at the same time, if you drink that tea, you get energy, you feel fresh because of the antioxidant effect, whatever. At the same time, you keep on going to toilet, okay, because of the diuretic effect. That is because of the complex mixture of bioactive compound present in the extract. Okay. Uh, so the product, it can be liquid or in some semi-solid state or in dry powder and are intended for oral or external use. Okay, depending on what research you are doing. For example, if you are trying to find a skin whitening effect of a plant that is for external use, normally you try to find something semi-solid, or if you have a dry powder, you have to prepare it in a cream form for external use. If it is for treatment of colon cancer, for example, it need to be used orally. Okay, if you are doing on diabetic, then it is normally for you to give orally or intravenously. Okay, so whatever the extraction procedure that you tend to choose later, you need to know first what is your target. What type of bioactive compound that you are going to isolate for what type of disease. Okay? So this you have to discuss with your lecturer in your final year project. You have to read a lot on other medicinal plant paper, journals to get the idea. Okay? Some people they like for example, Dr. Roslida, she used to during her extraction, most of the time she tend to use hexane. Okay, hexane to get the lipid soluble compound. Okay, for her research. So for chemists, for example, chemists they don't like to use hexane because hexane is highly toxic. Okay? But for the sake of research, you can use it to isolate the compound for knowledge. Okay? And still you can develop it into a product, into a drug, but it have to go through several other procedures lah. So if you work on medicinal plant, normally you will not get your product published or a product released for sale in only 5 years. Normally 10 to 15 years only you can get a product that is non-toxic, that can be used by people all over the world. Okay, so it requires extensive study, in-depth study for you to get the end product, which is a drug or medicinal product that is effective but non-toxic to for human consumption. So medicinal plant constitute an effective source of both traditional and modern medicine. 80% of population have been known to use medicinal plant, especially in rural area, whereby there are middle income to low income people, especially for example in Africa, they tend to use 80% of the people in Africa, they tend to use more people in Africa actually reliable on medicinal plant. They choose medicinal plant as source of medicine to treat their disease instead of modern medicine, because this is very expensive. So, and then medicinal plant, why you study medicinal plants? They are richest bioresource, okay, for drugs of traditional system of medicine. They've supplied drugs, new drugs based on traditional system of medicine. For example, you have Malay traditional medicine, you have Chinese traditional medicine, you have Indian traditional medicine, Arab traditional medicine. All of these can be used as your first source in your study if you are interested to study medicinal plants. Okay. You need to find what plant. For example, you can ask your grandmother, you can ask old people what type of plant is used for what type of disease. Then you can propose to your supervisor, or you can use the plant that your supervisor has been working with, and then it is source for modern medicine. For example, morphine. The direct example of modern medicine is morphine and aspirin, acetylsalicylic acid. They came from medicinal plants. Okay. They are also a source of nutraceuticals, food supplements, and then they are used as in folk medicine as well. And they are also used in as a pharmaceutical intermediate, and also as a source to produce synthetic drugs. That's why you have from morphine, you have heroin, you have codeine. That those are synthetic drugs that are produced based on the chemical structure of morphine. Okay, you have fentanyl. Okay, those are synthetic drugs. So all these came from medicinal plants. Okay, so if you can find, as I told you earlier, any plants that can help to make body sador, muscle body like this, you can be a rich person. Uh, medicinal plants, when you want to do extraction, the first thing that before you can isolate a compound, before you get a bioactive compound like morphine, for example, or acetylsalicylic acid, for example, you need to do extraction. You need to do preparation. So there are various type of preparation, okay, called decoctions, infusions, fluid extract, tinctures, pillular extract, or powdered extract. So we will look into this later. Okay. Uh, the name of the different name is actually because of the method of preparation. Okay, sometimes you use cold water to soak your leaves or plant sample, or you use hot water, and how many hours you spend to you leave your extract, you leave your plant in the hot water or cold water. That is that the that make the term different, no? You have decoction, infusion, tinctures. As I told you, tinctures is actually related to alcohol, meaning to say you soak your plant in alcohol. That is called tinctures. The evidence of ethnopharmacology. What is ethnopharmacology? Study on medicinal plants, the medicinal potential of medicinal plant or plants. So evidence of ethnopharmacology can be obtained from three main source: the Materia Medica, Historia Plantarum, Species Plantarum. Okay. So these are the books that can be used. But sometimes also you can base on your old folk beliefs, in your community, for example, in certain village, the people there, what are they using to heal fever, to cure fever, to reduce high body temperature. So they soak something into some plants into something, into water, and then they ask the patient to drink. So what is the plant? You can also get information from there. Okay. Normally this kind of books they covered most of the plant in the western part, no. Sometimes the Chinese medicinal plant, Indian medicinal plants, you need to get from the source directly from the Indian book, Indian Ayurvedic book, Chinese TCM, Chinese traditional medicine book. You can get information from there. But these are the main that have been compiled for your study. So when you do extraction, you have to standardize the extraction procedures. Okay, meaning to say why you need to standardize, so that when you do the extraction today, you do the extraction after 1 week, you do the extraction after 1 year, you do the extraction after 10 years, you will get the same amount of compound. Let's say you have compound A, B, C in the extract. So you want to get, let's say you do for the first time, you get 5% A in your extract, 5%, 10% B in your extract, and 7% C in your extract, the combination. So when you do it at different other times, okay, and on other occasion, you will get the same 5% A, 10% B, and 7% C. Okay, that is what we mean by standardization. That means also you need to maintain the time when you collect your plants. When is it? Is it on December, every December, or December to January, July to May, or only in July, only in February, every year? Okay, or during the winter time, something like that. You have to standardize. And which part of the plant that you are talking about? The leaf, the bark, the stem, okay? And then how long will you be soaking the plant in your solvent? What type of solvent will you be using? Is it water? You cannot do extraction sometime in water, sometime in ethanol, because it will give you different type of compounds. Okay. For example, ethanol, although it is water soluble, it is also lipid soluble. So when you do extraction with ethanol, you tend to take out all the polar and non-polar compound into your extract. If you use water, you will only get the water-soluble compound. So you leave the non-soluble, the lipid-soluble left in your leaf, in your sample. Understand? So that's why you need to do standardized extraction. Okay. So the solvent is called menstruum. Okay. When somebody tell you talk about menstruum, that means solvent used to soak the plant sample. Okay. So once you have standardized your method, you can use the extract as your medicinal agent. Okay. That's why if you read the journal, there are material and method section. You have plant extraction section, whereby you have to describe the plant was collected between November to December 2001. Which part was used? How was the extraction done? In the ratio of what? How many gram of plant and how many ml of water was used? Let's say, for example, 1 to 20, 1 to 10. Okay. How many days were the plants soaked? How many hours? How many minutes? Sometimes when you do extraction with hot water, you can soak for 25 minutes. After 25 minutes, you filter the sample, you take the supernatant, your extract, and you can use it already. Okay, for testing on animal or in vitro, in vivo or in vitro. Okay. If you use ethanol, you can use it as directly as tinctures. Okay, you can give to, if you want to test on animal, you can give it orally to the animal. Okay, so if you use water, you will get a fluid extract. So sometime when you do water extraction, after you get the supernatant, what you can do is you can remove the water using the procedure called freeze dry. Okay, whereby you take the water extract after you have filtered the the, the plant and the solvent. You take the extract, you kept in a minus 80-degree freezer for 48 hours, the shortest time, or 1 week, whatever you decide. After that, you get an ice, a solid ice. So you put this in a freeze dryer machine. So what happened? The freeze dryer machine will suck out the water in a vapor form, because you know the ice, it tend to release vapor, right? When it melt. So that water is sucked in a vapor form. So what is left is the brownish color. The what is left is your extract. Your crude extract will be left. The water will be sucked out into the machine and released from the machine. So what is left is your extract. Okay, so that crude extract in a powder form, you can use it to prepare tablets or capsules. You can use it if you tend to give it orally. If you want to treat a disease, for example, you want to treat diabetic on rat, you want to induce diabetic on rat, and you want to see whether your plant extract can cure diabetic or not, you can prepare it in tablets or you can give it in a fluid extract depending on your research. Okay. Okay, please answer his question. Any question?
No.
No, doctor.
Clear?
Mm,
All right.
alright.
Okay, as I told you just now, there are various techniques to do plant extraction. You have maceration, infusion, percolation, digestion, decoction, hot continuous extraction, or using chocolate apparatus. Okay, okay the first the first seven, okay, until aqueous alcoholic extraction by fermentation. These are normally the traditional method. Okay, these are just normally called traditional methods. The rest are the modern methods using certain machine, uh, highly expensive machine, okay, which are not which are available only in certain laboratory. For example, in IBS, okay, you can find this kind of machine in Institute of Bioscience. Okay, but normally in our laboratory condition, we only use the traditional ways of preparing plant extract. As I told you, the different names is due to how you prepare the plants. Okay, we jump to certain, just to tell you what is maceration. Okay. So your whole or coarsely powdered crude extract, okay, allowed to stand at room temperature for a period of 3 days with frequent agitation until the soluble matters get dissolved. The mixture is then strained. The mug is pressed. So the solid part is then pressed to release all the supernatant to get all the liquids. So then you have to filter the liquid that you get. Then only you will have your crude extract so that you can remove all the small particles, That's why you need to do filtration. And infusion, you use a fresh infusion are prepared by macerating. It is a maceration process, but you use either cold or boiling water. Okay. So you have a dilute solution of readily soluble constituent of crude extract. So the supernatant is then ready for use as an infusion. But its digestion, it is a form of maceration also with gentle heat. Okay. For infusion, you use a boiling water. Okay, that is the difference. For example, what is infusion? If you use hot water, it is a boiling water. While in digestion, you use only gentle heat. Okay, during the extraction process. So sometimes you can use uh, you can use you boil a water, then you put cold water until the temperature became 80 degrees Celsius. Then only you pour the 80 degrees Celsius water into your dried plants, dried sample, the plant sample. So that is one example of maceration. You only give a gentle heat, not a boiling or 100 degree Celsius heat. Okay? So that is the difference. And then you have decoction. Okay, the crude drug is boiled in a specified volume of water, normally 1 to 4, for a defined time. You have defined time also. How many hours? Okay. For example, you put 1,000 ml of water against 250 gram of your sample, plant sample. Then you boil it. Okay, you give heat, you boil it until the volume reduced to 1/4 or quarter. 1,000, it become 250 ml. That is called decoction. Okay, so you have also percolation. So normally the percolation was prepared by mixing the plants with specified menstruum, the solvent, and allowed to stand for 4 hours in a closed container. Okay. After that, the sample or the supernatant was collected. So you can read all these various traditional methods. Uh, I just want to tell you that the the modern method. Okay, we will not go detail on the modern method. It's just for your information, because in the end, when you do your research, you will use only the the traditional method. Okay, you don't have to go through detail on the machine that is used to do counter current extraction and so on. So this been covered. So these are some of the technique used for aromatic plants. Okay, so some you have plants. Sometimes you are looking for aromatic plants. You want to isolate the essential oil, for perfume preparation of perfume. So this is some of the method used. But when you are doing on medicinal plant, normally you use the other type of extraction. Okay. Okay, choice of solvent. Successful determination of biologically active compound depend on type of solvent. Okay. You if we, I think you know that plant there are various type of bioactive compound which can be classified into, for example, flavonoids, tannins, saponins. Okay, so this type of triterpenes. So how do you isolate each of these compound specifically? That depends on the solvent that you use. Okay, some compound they are naturally lipid soluble, so you cannot use water. Okay, you cannot use water to extract them, so you need to use solvent. And solvent also they are polar until non-polar, a series of solvent. If you list them from polar to non-polar, they are intermediate solvent, whereby they are solvent that only isolate non-polar compound. There are solvent that can isolate both polar and non-polar compounds. There are solvent that only isolate polar compound. Okay, so depending on the solvent, the choice of the solvent is influenced by what is intended with the extract, what you want to isolate. So that determine the choice of solvent. So if you look at this part, water normally, if you want to, for example, or this I give you one example. If you are working with antimicrobial activity, so water is the best solvent, okay, to isolate the bioactive compounds, because mostly water soluble flavonoids or anthocyanins, they don't have antimicrobial significance. So you don't want to isolate this kind of compound. Okay, you want to isolate water soluble phenolics that have antioxidant activity. Okay, so if you use acetone, it dissolves hydrophilic and lipophilic. This is intermediate solvent, also used for antimicrobial studies. Okay. So if you look at the slide, you can see aqueous acetone is more better than aqueous methanol in extraction of tannins and phenolics, which are good antimicrobial agents. Then you have alcohol, methanol. Methanol is cytotoxic in nature compared to ethanol. So if your extract is you targeted for consumption by human, okay, so you need to use ethanol, even though methanol is cheaper than ethanol, but the consequences of consuming methanol-based product is more hazardous than ethanol. Ethanol is safe, okay, highly safe for consumption. Then you can use chloroform if you are targeting to isolate tannins or terpenoids, or you can use ether, a more non-polar compared to chloroform in the list. Then you have dichloromethanol if you are targeting only terpenoids. So once you use dichloromethanol, you know already you are targeting terpenoids. You will not get flavonoid, you will not get tannins, you will not get saponin, you only will get terpenes. Okay? So these are the main solvent normally used. Sometimes people use butanol and so on, but these are what that is covered under alcohol lah. Petroleum ether, ether is under non-soluble solvent or lipid soluble solvent. Okay. Okay, what is the properties of a good solvent in plant extraction? A properties of good solvent in plant extraction, it must have low toxicity. Okay, it have ease of evaporation at low heat. It can evaporate easily at low heat. You don't need high heat, because if you use high temperature, you heat to evaporate the solvent, what can happen is that some of the bioactive compound that are not stable at high temperature, they can denature or destroy. So you don't want that. So what normally when you do your extraction, you have to consider evaporation at low heat. Okay. And then promotion of rapid physiologic absorption of the extract. So absorption of rapid absorption. It can promote rapid absorption, meaning to say the extraction of the bioactive compound out of the plant sample can be done rapidly. Okay, it have a preservative action. It can preserve the bioactive compound that have been isolated, and so that it is not destroyed. Okay. Then inability to cause the extract to complex or dissociate. So it can prevent the extract from dissociate, denatured, and so on, okay, or form a complex with other compounds. So it can prevent all this. Sometimes some bioactive compound, they tend to have double bonds, they tend to have charges. So what happen? They will try to bind to something and form a complex. Or because of the charges, they tend to dissociate, denature. So it become inactive. So what we need is a solvent that can prevent this, prevent the active compound in the extract from producing a complex or compound or dissociate to become inactive. So this 5 is the properties of a good solvent. So this is type of type of solvent that can be used to extract different type of bioactive compounds. Okay, depending on what you are targeting, you can choose one. Okay, for so let's say for example you are using ethanol or methanol, but when you do extraction, you will get all this in your sample. So what you need to do, there is from extraction, what you need to do after you get a crude extract that contain all this. Let's say you want to take only polyphenols. What you need to do after extraction is you need to do partitioning. Partitioning means you use solvent that have polarity maybe lower than ethanol or higher than ethanol to remove other compound that are present in the ethanol extract, so that whatever, so that you can get only polyphenols. Okay, that is called partition. Sometimes partitioning is not enough, then okay, sorry. Sometimes after you do the partitioning, you want to know what is the bioactive compound. Let's say you have the extract rich in polyphenol after you do the partitioning, what you need to do is to do fractionation. So you have 3-stage extraction, partition, and then fractionation. Okay? And the last step, number 4, is isolation of bioactive. Then only you can do isolation of bioactive compounds. So the procedure is very extensive. It is normally a PhD process, a 3 years process. If you do a PhD, you can do isolation of bioactive compound from zero, from crude extract until you get the pure compound. So like in my case, normally when I do crude extract, I will do ethanol as a crude extract. I get ethanol. From there, I will do partition, whereby first I will mix the ethanol extract with water, and then I will shake it so that everything is mixed up. Then I will put petroleum ether, which is non-polar. Water is polar. Then you put petroleum ether in the mixture. Petroleum ether there will be 2 layers after that, because petroleum ether and water, they will not mix together. So you will have a water-based compound, and you will also have a lipid-based from petroleum ether compound. You can separate them two. Then you put into the water mixture again. You put what we call it, ethyl acetate. Ethyl acetate is similar, almost in one group. In acetone, they are called intermediate solvent because they can bind to polar and non-polar compound. So when ethyl acetate is put into a water mixture with ethanol, there will also be a 2-layer form. So you take the ethyl acetate layer out, then now you have 3 type of extract already. You have a water-based extract, ethyl acetate extract, and petroleum ether extract, which represent water is a polar, petroleum ether is a non-polar, ethyl acetate is a intermediate polar, meaning a compound that have a combination of polar and non-polar compounds. So from there, we can test back which of the extract is the most effective extract for certain disease. Let's say diabetic, for example. Let's say you find out the petroleum ether part is more effective, then you can proceed the petroleum ether extract with fractionation using column chromatography that you have learned. Okay, you use column chromatography, so you get all the band that you learned already. You collect the band, you test each of the band in the animal again, see which band help to reduce diabetes effectively, then that band is used for isolation of bioactive compounds. I hope you understand what I'm trying to say. Any response?
Yes, betul.
Can understand or cannot understand? Please ask.
Can, Doctor.
Can I? So
Doktor,
the slide,
doktor,
yes.
boleh tak doktor repeat dekat partitioning tu guna apa? Solvent yang lower polarity
Which one?
Dekat partitioning punya step.
Okay, okay. When you do extraction, first you will get a crude extract, right? You will use, let's say you use ethanol, you use methanol, or let's say if you use water and chloroform, they are both polar and non-polar, so you will directly get a specific type of compound already. But if you use ethanol or methanol, you will get polar and non-polar compound. So what you need to do is you have to do partition. Okay, you have to do partition. So what you need to do is when you use methanol or ethanol, you need to first isolate the compound into polar, non-polar, and intermediate. So what you need to do first, when you have a crude methanol or ethanol extract, you dried it, you have a sticky paste of crude extract, you put water inside it, you put water, you shake it until everything dissolve. Okay, you will have some part that are not dissolved. That is a lipid based part. So don't worry. What you need to do after that, you put petroleometer also in the mixture. Then you shake again. So what happened? The non-soluble part, when you put water, that non-soluble part will dissolve in petroleometer. But because petroleometer and water, due to the presence of water, they will not mix. So what happened? They will be separated into two layers. Okay? So you take out the petroleometer part because there is a non-polar part, but in the water part, there are still intermediate and polar, highly polar and intermediate polar compound. So what you need to do, you put ethyl acetate into the water ethanol or water ethanol mixture, the earlier part. After you take out the petroleometer, you put ethyl acetate, then you shake it for several times, and then you leave it for 1 hour or 2 hours, and then you will get another 2 layers. You take out the ethyl acetate part, then you have 3 already. You have a water-based part, which is polar. You have a ethyl acetate part, which is intermediate polar. Intermediate polar means they are polar and non-polar based reactive compound. And then you have the earlier part, the petroleum ether part, which is a non-polar compound. Okay.
Okay, thank you, Doctor.
Alright. Right. So I will skip the slide water until I explained already. Okay, I move to slide 23. Okay, step involved in plant extraction. These are the steps: size reduction, extraction, filtration, concentration, and drying. So these are the machine. The top one is the rotary evaporator. The purple color is actually your extract. It is not actually purple color, is just for your reference. Okay, the purple color is a place where you put your extract, your supernatant without the plant residue. After you filter, you remove all the plant part, the leaf or whatever. You take the supernatant, you put in a beaker like this. You attach to the machine. Then what happened? You inside this part, you have a water, boil water at whatever temperature that you decide, 80 degrees, or if you want 100 degrees, you want to prepare infusion, decoction or whatever. Depend on the water temperature, you can set the temperature here. So what happened if you, after you heat it, this solvent, if whether it's water or alcohol, especially alcohol, alcohol or chloroform, organic solvent, sorry. The organic solvent, after you heat it, it will become vaporized. The vapor will go up, and it will go inside here. This blue color is a cool water. Okay, inside the coil there is cool water. So this cool water will make the vapor turn to water again. So what happened? The water will fall and collected into this yellow color part. So whatever, after all the crude solvent have been vaporized and collected in the yellow color part, what will be left is a sticky crude extract, your extract. This is what you want. Okay, this crude extract, if it is methanol or ethanol, you can do partition like I told you just now using pet ether. You mix pet ether, then you boil again. You remove. You put pet ether. You separate it, and then you put acetate, and then you separate it. You have 3 extract. The pet ether and the ethyl acetate part, you can use rotary evaporator to remove them to collect your specific extract. While the water-based extract left, the third one is what you need to do is you need to put in a minus 80 degree temperature. Then it become harder at ice. It become an ice. Then you put in the bottle, and the water will be vaporized and sucked by the machine, leaving only your dry water extract. No more water, only the dried powder extract. So this is a freeze dry machine. If you go to our pharmacology and pharmacology and physiology lab, we have these 2 machine. Okay. So these are the machine used, important machine lah, if you want to do medicinal plant extraction. Okay. When you do extraction, the size reduction, meaning to say if you have a leaf or you have a bark, you have a stem, or you have whatever sample part from the plant, you need to grind it to become powder, whether powder form or coarse. Coarse mean like a tea. If you open the sachet of a tea, you pour it, the leaf is, you can see a shape, what they call it. It is not a fine powder, like it is coarse, kasar. So you can have to grind it first, and then after that, you do the extraction. You have to know which solvent are you going to use, what is the ratio. Like for me, if I'm doing my study, I use 1 to 20, 1 gram of my sample to 20 ml of solvent, so that every part of the sample of the 1 gram sample is covered by 20 ml of my solvent. Okay, if I use 10, then some part will not be covered by the solvent. So that's why I standardize my. If you read my paper, since I start until today, I use 1 to 20. So what part for myself, for example, since 2003, 2001, I've been doing on the leaf. So you can imagine you have a plant, okay? You did not, after 10 or 16 years, I'm only studying the leaf. I did not study the fruits, I did not study the flower, I did not study the bark or other part of the plant. So you have a lot of plant that you can do exploration, other part of the plants. So after that, after you get your extract, you have to filter them using filter paper. Then you also can use cotton wool first, and then you also can use vacuum pressure. Okay. First, normally, like for me, I ask my student to first use. You have the funnel. You put cotton wool, then you pour your extract, and whatever pass through the cotton wool, there will be small residues of the plants. Then only after that I will use filter paper. Then after I use filter paper, still inside the extract, you some of the part that are not filtered out. What you can do is you use vacuum pressure. Okay, you use vacuum to suck out your sample, leaving all the minute residue left, not taken up together with your extract. Okay, so And then after filtration, you do the concentration. This is what we mean by concentration. You use rotary evaporator. Sometime when you want to, if you want to expose to temperature, high temperature, let's say 100, then you don't have to use reduced pressure. But sometimes you want to save your compound from heat effect. You want to prevent the loss of some of the bioactive compound that are easily denatured by high temperature. So you use reduced pressure. You attach some one equipment, a vacuum pump. So this vacuum pump will induce vacuum pressure inside this rotary evaporator system, whereby you don't have to use high temperature heating to vaporize the solvent. You only use 40 degrees Celsius of heating. You heat the water outside here at 40 degree, but the solvent inside here, whether it is methanol, ethanol, chloroform, whatever, they will be vaporized at 40 degrees Celsius. If you did not use reduce pressure, or you did not attach the vacuum pressure here with the machine, what you need to do is, for methanol, I think you need to heat it at 80 degrees Celsius. Okay, I think most of the solvent will be vaporized at 60 to 80 degrees Celsius. Okay, so by using reduced pressure, using vacuum, you can reduce the temperature to 40 only. So you will not expose your extract, the purple color part, to temperature that is higher than 40. So meaning to say, your compound inside the extract will not all destroyed. Okay, completely not destroyed, because at 40, mostly plant can still, the compound can still be stable at 40 degrees Celsius. Then after this, you need to do the drying process. So for water-based extract, you would use freeze dryer. This is a freeze dryer. Okay, you dry it to become powder, or you drying it using the same equipment, the rotary evaporator, until no more solvent is being collected inside the yellow color part flask, and it is considered complete drying. Okay. Also, you can use a drying chamber. It is sort of a chamber, almost similar to the one that you use in a microbiology lab, or when you do the chamber is, example is when we do the practical class for column chromatography, at the back of the lab, that is the drying chamber. You can turn on the machine, so it will give a flow of wind. Okay, that flow of wind can help to dry your extract that you put inside there, provided you cover it with a glass. You close the section with the door, the windows. You close it, then you can dry your sample using drying chamber. So there are 3 methods of drying your extract. Why you use drying chamber? Sometime when you do evaporation process, until the last step, whereby there is no more solvent that can be sucked up by the system and collected in the flask, yellow flask. What you get is your sample is sticky, and they are not solid. They are semi-solid. They are still in a liquid form. So you cannot do drying using rotary evaporator anymore. So what you need to do is, you put your sample in the drying chamber. You open the cap of the bottle, or you covered it with aluminum foil, but you make a hole on the top of the bottle so that the remaining residue of the solvent can be dried in the drying chamber. Okay. So this one you just read through. I just explain to you the size reduction. Okay, earlier we have talked about this. What is extraction? You can have percolation, cold aqueous, decoction is hot extraction. Okay, and then solvent extraction, it can be cool or hot. Okay, this one for aromatic plants, we will discuss about it later. So when you do extraction on aromatic plants, you will have 5 different type of product. Okay. So that one will be covered later, and now we move on. Any question?
No, doctor.
Should we stop and continue later and tomorrow?
So,
How? Boleh, tomorrow lah.
what time tomorrow?
10 or 11? How?
We got quiz tomorrow, doctor.
Oh yeah. Then the day after tomorrow, nak habiskan hari ni. Sure, more than 2 hours. Boleh ya? Ada half lagi lah. Baru 27, ada 78. Okay, you all decide. Before that, I'll continue. Okay, parameters influencing the quality of an extract. Okay, there are several parameters that affect the quality of extract. Okay, first, the plant part that you use as a starting material. Whether you use the leaf, whether you use the bark, whether you use the stem, or any part, the fruits of the plants. Okay, that will affect the quality. So what you need to do when you do medicinal plant research, first, you need to know what is the traditional usage of the plant extract. Normally, the traditional people, traditionally, the people use how did the people use the plants? Okay, whether they put in hot water, whether they use the leaf only, whether they use the fruits, whether they use the bark. If they use the bark and you take the leaf, then you will not get the quality extract. Okay, you might have the same effect, but the effect might not be as good as the one that is traditionally used. So you have to know which part is actually being used traditionally. Okay. For example, to do aromatic plant extraction, you want to take the plant that can produce a good smell extract, right? So, for example, pokok gaharu kan. gaharu, what they call, I don't know, in India, they call it oud. It smells very nice and very expensive. Gaharu is very expensive. Okay. Some tree can be up to 80,000 after 15 years. If you grow it now, after 15 years per plant, for per gaharu trees, you can get 80,000. Okay. But get ready with people from outside, no? They might tend to come and chop out the tree and take it, steal it from you. Okay. And then the second is solvent used for extraction. Also will affect the quality. Okay. Sometimes the plant is effective, for example, to treat diabetic. I give example again, diabetic. Okay. But the plant, the nature of the bioactive compound that produce the anti-diabetic action is actually the non-polar bioactive compound. So if you do extraction using water, then you will not get the extract that produce a good quality anti-diabetic effect. Understand? So what you need to do is you need to isolate using the non-water soluble extraction. Okay. So for example, if you afraid to use chloroform, okay, you're afraid to use chloroform because chloroform is toxic, or petroleum emitter, any non-solvent, any organic solvent that are non-polar, they are highly toxic. Okay. So what you need to do is you use ethanol, because you know ethanol, it can take out polar and non-polar compounds. So what you need to do after that is you do partitioning. Okay. Then only you get the non-soluble, lipid soluble bioactive compound with high anti-diabetic effect. So solvent also play a role. And then the extraction procedure also will affect whether you leave the plant's extraction for 3 days or 1 day or 20, or 1 hour, for example. That will also determine the quality of the extract. Maybe some extraction need to be done 3 days to get the maximum amount of bioactive compound with anti-diabetic effect out of the extract. So if you do the extraction only for 1 hour, you will not get the maximum number of anti-diabetic compound to come out from the extract. So you will get less effect. Right? The quality of the extract will be lower. Okay, there is explanation for that. And then effect of extracted plant phytochemicals depend on. So once you have extracted the plant, you get the crude extract. The effect of phytochemicals inside the extract depends on nature of the plant material. Okay, nature of the plant material. What do we mean by this? Okay, some plant, they tend to produce compound A, which is a high anti-diabetic agent, during winter time, not during the snowy, cold weather. Sorry, during what? Winter snow lah. Okay, not during snow. Okay, during what they call it, the 4 season. Alah, tanya sekarang. Can somebody help? You know, you have 4 seasons. Okay. So sometimes some plants, they tend to produce compound with high anti-diabetic effect, for example, during one season, not the other season. So if you collect it during the season that it is not producing a high amount of compound A, for example, that produce high anti-diabetic effect, so you will not get an extract that is effective. Okay? That's why when you do research, you need to record when did you collect the sample, from which area that you collect the sample. Sometimes you collect it in Malaysia around UPM, around the vicinity or around the place around UPM. The result that you produce is remarkable. But when you collect it from somewhere from Perlis, the same plant you collect it from Perlis or you collect it from Thailand or you collect it from Sabah, the same plant will not produce the same effect. This is due to the nature, maybe the plant soil, whatever will give effect. So that is what we mean by earlier I said about standardization. Okay, where did you get your sample? You need to record everything. And when you become a researcher later, you need to apply for research grants, especially whoever that will do on medicinal plant. You have to remember one thing: when you make a proposal, the extraction have to be water or ethanol. Okay, you cannot say I want to my extraction will be carried out. The extraction will be carried out using methanol, using chloroform, using petroleum ether. Definitely, your proposal will be rejected. Okay, because you are using toxic compound. So this is the main issue. So when you do you prepare your proposal, you should say that you will use water or ethanol. Okay. So that is one of the tips in preparing your simple tips on what you have to write in your proposal. Okay. And then its origin, as I told you, whether it is from Kedah, it is from Thailand, it is from Africa, wherever the sample came from, you need to record it so that you know. Maybe you take the sample from India, okay, the same plant, you take it from India or you take from Africa, and you study it in Malaysia, you found out it is not effective or it costs only 30% of antidiabetic effect as compared to the same plant that you collect from Pahang, for example, from reserve forest in Pahang, Kuala Tahan, whatever, or you take it from Indonesia, which is more effective than the one that came from other parts. So the origin itself also will affect the phytochemicals. Okay, degree of processing, all the degree of processing, whether there is use of extreme heat or too long extraction, whether this will denature the phytochemicals or reduce the extraction of targeted phytochemicals, and then the moisture content. If too moisture, then you will get less phytochemical, for example, and then the particle size used, when you grinding, whether it is too small or too big. Okay. So this will all affect the extraction of phytochemicals. Okay, this one you just read selection of plants. Okay, which part that you will use. Then normally they are air dried until constant weight. So because you need to dry it, under shade. Okay? You dry it, air dried under shade. Normally, like for my student, or most of the student, they dried it in the lab, okay, laboratory lab. On the floor, they put a newspaper and put the plant on the floor. Or sometimes if the oven is available, then they can put in the oven at the temperature of 40 degrees Celsius. Okay. So normally for my student, they put at 40 degrees Celsius, or they put in the lab and on the floor for two weeks. I ask them to dry it for 2 weeks so that we get a constant weight. We remove all of the water, so that when we weigh the sample, it will have an accurate weight. Okay, berat dia akan sama, same weight. If it is 1 kilogram, it is 1 kilogram of sample, not because 1 kilogram due to the presence of water in the sample. So that's why you need to dry it until a constant weight is obtained. Filtration, as I told you, you use cotton wool. You can use also filter cloth. And then finally, you use filter paper, Wattman number 1 or Wattman number whatever it is, depending on your lecturer. Then finally, if necessary, you can use vacuum pressure, okay, to suck the extract. So this is an example of vacuum pressure, vacuum pump, okay. So this is a vacuum pump. You put your extract here. So the vacuum generator will suck your extract, pass through this filter, and you will get a clean extract inside this flask, which is ready for use. Okay, this is the final step, okay, that you can carry it out. So concentration, you need to concentrate your sample using rotary evaporator, or also you also can use chamber dry to concentrate until you get a very solid dried extract. So this is just for you to read. Drying, you need to use freeze dryer or rotary evaporator. So once you have a dry powder, you have to dilute them using suitable diluents, okay, before you can prepare different concentration of extract. Okay. So for example, if you are using water extract, after you dry using a using freeze dryer, you get a powder form of water extract. What you need to do, you dissolve it back in water. Okay. At certain, depending on the dose that you want to prepare, whether 50, 20, 50, 250, and 500 mg per kg. So you need to measure the dried powder and then you put water so that you prepare to the exact dose. Or you can also measure the dried powder if you want to prepare tablet directly. Weigh it, okay, you weigh the powder, and then you can use a specific machine to prepare a tablet or a capsule. Okay. And then sometimes you can use spray dry machine, okay, to get a dry extract. Okay, variation in extraction method. There are variation in extraction method, length of extraction method. As you go through some of the slide, you see some extracted for 4 hours, some are extracted for 3 days. Okay. Solvent use, it can be water or lipid soluble solvent, pH of the solvent. Sometimes this part, normally a chemist who are doing on medicinal plant, they are very, very particular. Okay, let's say you want to isolate one type of compound, one class of compound, let's say for certain type of acid-base compound. So you have to modify the pH of the solvent. You put some other reagent or chemicals that can reduce or increase the pH, so that when you add to the plants, you will get the exact bioactive compound that you want. This is a role of medicinal chemist. And then temperature, particle size of the plants, and the solvent to sample ratio. If the solvent is not enough to soak the sample, what will happen? If you have less solvent, then the solvent will become concentrated with the bioactive compound. Some part of the sample will not be able to be extracted by the solvent, so you will not get the exact or the maximum number of bioactive compound out from the sample, in a sense. So what you need to do is your solvent should be more than your sample so that it covered, but not that much. Let's say 1 to 20 for me is nice, but if I put 1 to 30 or 1 to 40, I am just wasting my solvent. Okay? So 1 to 20 is just nice for me lah, in my research. So the ratio itself also play a role. If less, then you will not get, all your sample will not get extracted by the solvent. Okay. Parameters for selecting an appropriate extraction method. There are parameters to be considered when you do extraction. So first, authentication of plant material by botanists. Every time you're doing research with medicinal plant, the first thing is to take your plant to a botanist for authentication. Because why? Sometimes you might say it is plant A, okay? But it is the, plant, there are certain varieties, okay? On the same species, there are certain varieties, different varieties. So you need to go to botanist to confirm whether you are using the exact plant, because sometimes also, if a plant is known to the Malay culture, for example, if it is known as senduduk, pokok senduduk, but in certain part of the country, even in Malaysia, some part, let's say in Indonesia, it is known with other name. Okay, so it might not, you might take back to your lab different type of plants. Okay, or maybe they are same name but different plants, can also happen. So because of the different culture and so on, so forth. So what you need to do is, the first thing is, when you have a plant, pick it back, bring to a botanist. You take picture first. If it is a big tree, you take a picture. You take picture of the tree, picture of the leaf, picture of the fruits, and try to bring back the leaf, the fruits, for example, so that the botanist can have a look and confirm what is the species. And then use the right plant part. The age of the plant also have to be considered. The time, season, place of collection. Okay, so all these part also have to be considered. I've explained this already. Nature of its chemical constituent, which chemical constituent that you want to isolate out. Okay. Grinding methods, powdering techniques, whether you need a powder form or a coarse form of your sample, or you need to, when soaking, you need to shake it or not. Okay, during soaking, or you just leave it on the table not shaking for 3 days, or you shake it for 3 days. The methods nature of the constituents, polar or non-polar, quantity of water or menstruum, the ratio, okay, the extractor fabrication, material and design also affect, okay, the design of the extractor, analytical parameters in the of the final product, final extract. So this is all the things that will affect, okay, the TLC, HPLC, whether they will be used analytical parameters of final extract, whether you will do TLC, HPLC or not. Okay, if you have a water product, then you do the analytical parameter using GCMS. It is not acceptable because GCMS is used for volatile compounds. So water-based extract is not a volatile compound. So if you publish a paper, you send a paper saying that you have water extract, and one of the analytical parameters that you use is GCMS, whereby you inject your water extract into the GCMS, you get this chromatogram. Okay, you show a chromatogram with several peaks and so on. It is not acceptable because normally water extract is not analyzed analytically using GCMS. They need to use HPLC or UHPLC. So this kind of thing also will determine the extraction method. If you are if you want to use GCMS, then you should know that it should be organic solvent extraction. It should not be water extraction. Okay, this is the normal lab for medicinal plants analysis. You see, this is HPLC. Okay, normally you have HPLC. You see GCMS. Okay, you have a complete set in one lab. Okay, this is a dream for a medicinal plants researcher. Okay, unfortunately, we don't have that. Even though we are RU. So this, as I told you earlier, you have traditional and modern method of extraction. I will not go detail on the modern one. Okay. Personally, I would like to tell you that I have never myself been really involved with using all this modern. It's just a method whereby you use specialized equipment to maximize the extraction. For example, you use microwave. The microwave is quite simple method, but it's still, I mean, it use additional equipment, okay, instead of the simple extraction procedure. So you put your extract in water, for example, or in solvent, ethanol maybe, and you put in microwave at certain temperature to heat the extract, because it is known that microwave effect, it can trigger the bioactive compound to change its structure and become more active compared to the normal condition, because of the heat effect on the bioactive compound. Some of the bioactive compound tend to change their structure, and what happened is that they become more active. Maybe because the site, active site, you learn this already during pharmacology. The active site for chemical to bind to receptor, by using heat, maybe the active site is open permanently. So when this extract is given to a plant, to an animal that have diabetic, for example, so because of the active site is open, it bind to it bind to the receptor, the respective receptor, and it prevent diabetic to occur. Okay, it stimulate the part that produce insulin to keep on producing insulin until the blood glucose level reduce. Okay, that is one example lah. Also, you can use ultrasound or sonication. Okay, this kind of extraction, you give sonication. Basically, you give sonication to the extract. You put the extract in like water bath. Then you sonicate it. Okay, you sonicate it, and what happened during sonication? It will push out every bioactive compound in your plants to get out from the plant and go into your solvent, okay, whether it's water or organic solvent. So you get maximum extraction. So that, and then supercritical fluid extraction, for example, you can use carbon dioxide, okay, in liquid form, or you can use nitrogen liquid. Okay, normally we use carbon dioxide, okay, in a fluid form. You do extraction. So when after you do extraction, you release the carbon dioxide into gas form. Then what is left is your dry extract. Okay, this is a safe procedure, but it is quite expensive. It's a quite expensive procedure. Phytonic extraction, you use new type of hydrofluorocarbon. Okay, all of this is actually the modern method is actually to improve the traditional method. Okay, actually it is used to improve the traditional method to get maximum yield of bioactive compounds. Okay, this one I've gone through already. And then sometimes you can use also a combination of water with alcohol. Okay, especially in Ayurvedic, they tend to do extraction whereby alcohol and water is combined together. Okay, you read on this just for your information. Okay, this is the big scale process. Fermentation, big scale fermentation process. So these are explanation for some of the modern methods. You can read through. Okay, I think I stop at aromatic plants. In 2 days, we try to finish another 18 slides because I myself cannot go on anymore. Okay,
Okay, doktor.
2 days from now, tomorrow, the day after tomorrow at 11. Okay, any question?
No question.
Tak ada?
Tak.
Okay, kalau tak ada, dismiss lah. Okay, bye-bye. Assalamualaikum.
Salam.
Thank you, Doctor.
Welcome.